So the next segment we are looking at, or compartment, is the stomach. In the stomach you find gland pretty much throughout the entire organ. We can distinguish between the cardiac glandular region about here, then we have the oxyntic glandular region pretty much to the entire body of the stomach. And then in the pyloric antrum region we also find secretory cells. There are a number of, you already know that from anatomy and histology, there are a number of different secretory cells in the stomach. So we have these mucus-producing cells, mucus neck cells, glandular mucus cells. There are different names for or describe the group of cells that overall produce mucus and bicarbonate. We get to this a little bit later. The bicarbonate, as I said, is required or is used to provide a very thin layer across the epithelial cells so they themselves do not get damaged by the massive acid production in the stomach. So the parietal cells are the ones that produce hydrochloric acid, the acid secretion in the stomach, but they also produce this intrinsic factor, which is another protein that is required or is used for protecting vitamin B12 from degradation in the acidic environment. So we have these R proteins and we have this intrinsic factor. Both serve the purpose of protecting vitamin B12 from premature degradation before it actually can be absorbed in the ileum. The chief cells, on the other hand, are those that produce digestive enzymes, the pepsinogen and the gastric lipase. So now we already have two different lipases, or two different sources of lipases that are acting in the stomach. The lingual lipase that we are swallowing, and the gastric lipase that is generated and released in the stomach itself by the so-called chief cells. I mentioned these two other names in parentheses, oxyntic cells and peptic cells. These are older terms, but you find them in textbooks. So you know what we are talking about. I won't use them in the upcoming discussions, so I will always refer to them as parietal cells and chief cells. Then we have neuroendocrine cells, the G cells producing gastrin, an important hormone that regulates secretions, D cells, somatostatin, and then these enterochromaffin-like cells, which we will also further discuss because they are very important to stimulate acid secretion in the stomach. We get to this in a second. So digestive functions, well, they produce the gastric secretions, contribute digestive enzymes. The pepsinogen, that's an endopeptidase, so it cleaves amino acid bonds. It's an endopeptidase, so this means they are attacking chemical bonds within the peptide, within the protein, not at the two ends of the protein. That would be exopeptidases. And for digestive functions, of course, the production and secretion of hydrochloric acid. The environment in the stomach lumen is very acidic. It can be a pH all the way down to 2 and even lower. And remember, pH is a logarithmic measure or a number. So a change, you know that, but I repeat it anyway. The change of 1 pH unit is actually a 10-fold change in concentration. So if you go from a pH of 7 to 2, you have 5 orders of magnitude changes in concentration. That is huge in terms of how much acid has to be produced to get an average pH in the lumen of the stomach. Now, why is it so acidic in the stomach? Well, there are a couple of reasons. First of all, the acidic environment is antibacterial. There are not many bacteria that actually survive an acidic environment that has a pH of 2 or 3. So that's a very strong antibacterial or protective mechanisms or contribution of the gastric secretions. There is an exception to that, and we will talk about that briefly in terms of when some of these protective mechanisms break down. I mentioned the R proteins, an intrinsic factor that protect vitamin B12 from being degraded, and therefore aids its absorption at the right site, which will be later on in the terminal ileum. But the main thing is the gastric acidity kills bacteria, and also it softens food and helps the digestive process and helps the digestive enzyme to actually chemically break down these macromolecules. And I mentioned the mucus and bicarbonate that provides this protective layer just at the apical membrane of these epithelial cells in order to protect themselves from, in quotation marks, self-digestion within the lumen of the GI tract. Pepsinogen, that's now the first digestive enzyme we encounter during or in response to the ingestion of a meal. That's an endopeptidase, as I mentioned. How is it activated? Well, first of all, the pepsinogen is a precursor form that is released in an inactive form. Then this low gastric pH converts this proenzyme pepsinogen into the active pepsin, and then pepsin itself can proteolytically cleave other precursors forms proenzymes, and therefore there's a positive feedback that then increases the concentration of active digestive enzyme, the pepsin. Pepsin works best in a pH around pH 3. It has a relatively broad pH dependence, so even if your pH drops to 2 or something like that, it's still going to be very active in breaking down the nutrients. Pepsinogen is stored in zymogen granules and is released by exocytosis. What stimulates pepsinogen? Well, the hormones. There are a number of hormones that stimulate this peptidase. Actually, all our 4 most important hormones can contribute to that. Stimulation by the parasympathetic using acetylcholine as a neurotransmitter is a strong stimulant for pepsin secretions, but there is also an influence from the beta-adrenergic agonists. And as I mentioned, the acid itself is a stimulus for pepsinogen secretion. The composition of the gastric secretions is dependent on the rate of secretion or release of secretions, and if you look at the acid itself on the basal rate, there's a relatively low release of acid, but if you stimulate the glands fully, you get up to 40 milliequivalent protons per hour under physiological conditions. Patients with, that develop duodenal ulcers tend to have a higher release rates of protons. And why does it cause the duodenal ulcers? Well, there's more protons produced, and the mechanisms that neutralize the gastric secretions that enters the small intestine are not keeping up. The pH drops also in the small intestine, and then eventually this can lead to the development of ulcers. I show you then a picture about that. At low flow rates, it's hypotonic, so it's less solutes per volume compared to the plasma. We always compare to the plasma composition. If at full rates or maximum rates, it's nearly isotonic with the plasma, and you can see from the composition here, it almost completely or exclusively contains or consists of hydrochloric acid. Now, this and a little bit later in the pancreas, I show you 2 cellular schematics about the mechanisms that are involved inside these parietal cells here. I'm not going to show you many of these throughout my lecture, many of these mechanisms, slide of cellular mechanisms, but this one you should remember because it has some direct ramification of some of the therapeutic interventions that can be applied to control excessive acid production in parietal cells. So what I want you to keep in mind is how these protons are generated in these cells. Basically, CO2 is always produced in cells through metabolism. Water is always present. These two compounds, CO2 and water, are at equilibrium with carbonic acid. And this enzyme, the carbonic anhydrase, is an enzyme that catalyzes and accelerates the production of carbonic acid. Now, this reaction would happen spontaneously. It's at equilibrium. But by having this enzyme, you can massively accelerate this reaction and therefore increase the rate of proton generation. So carbonic acid is at equilibrium with bicarbonate ion and protons. The protons that are generated by this pathway are then transported into the lumen of the gland and therefore ultimately into the lumen of the stomach. This occurs in exchange to potassium ions. This is an ATPase, so this is an active, primary active transport mechanisms that uses ATP in order to transport the protons out of the cell, out of the cytoplasm, into the lumen of the gland and the stomach. Why is this important? Well, as I said before, the pH in the stomach can be very acidic. So you still can produce and add more protons to the lumen, which means you have to transport proton against a huge gradient uphill. And this can only be achieved under usage of energy, and that occurs by burning ATP in order to fuel this ATPase and exchange protons against potassium, which are present in the lumen. Then, when you accumulate protons in the lumen of the gland, chloride follows, so you have electroneutral conditions. So that's why we have hydrochloric acid in the stomach. The bicarbonate that is also produced for each proton is then transported out of the cell at the basolateral membrane in exchange to chloride, which provides the supply for the chloride that is transported into the lumen of the gland. So if you stimulate these glands fully and you have a large amount of proton production and delivery of protons into the lumen of the gland. At the same time, you have a lot of bicarbonate production, which then ends up into the bloodstream that leaves the stomach. And if you measure the pH in the blood from the stomach after full or during full stimulation of the gland, you will measure that the pH becomes alkaline, and that's referred to as the alkaline tide. So this pH from that particular blood source source would be somewhat more alkaline compared to the overall pH in the blood because of this massive transport of bicarbonate into the blood that leaves the stomach under these conditions. Now, the reason why I bring up this overall mechanism is we have rather efficient medications to curtail excessive proton production in the stomach and therefore acid production. And these are drugs that specifically block this ATPase. Prilosec is one of these earlier ones, that there are different ones available of these proton pump inhibitors. But those are very efficient drugs because they bind irreversibly to the pump molecule and block it through this binding. They are efficient because they are irreversibly binding, so the only reason or the only way this blockage of the pump gets relieved is by actually exchanging the pump molecules with new synthesized pump molecules, which occurs all the time, but it takes a little bit of time, and therefore these effects of these medications are relatively long-lasting compared to other medications. So those are medications for treatment of reflux disease or ulcer disease that are widely used and fairly efficient in achieving this purpose. Control of gastric secretion, stimulation, so neuronal control. We have parasympathetic innervation that leads to hydrochloric acid secretion, but also pepsinogen, so the proton component, and mucus. Hormonal, histamine is a strong secretagogue for parietal cells, for acid secretions. It's released from these enterochromaffin-like cells that are found in the gastric mucosa. So they're the same location as the glands themselves, so this is a paracrine effect. Gastrin from the G cells stimulate hydrochloric acid secretions, and secretin is mainly responsible, among the hormones, for stimulating pepsinogen secretion. And also distension of the stomach, of the duodenum, at least initially, stimulates the gastric secretions in general. So most of the agonists or the mechanisms that stimulate the gastric secretions affect both parietal cells and therefore acid secretion, and also the chief cells, which mainly produce or are responsible for the release of pepsinogen. So the rates of the secretion of acid and pepsinogen is fairly well correlated. So if one goes up, also the other one goes up, and makes perfectly sense because you need the acid for the pepsinogen to be cleaved and become active, but also to be able to work in a appropriate environment. There is an exception to that. Secretin actually is more specific towards stimulating pepsinogen secretion, but is actually inhibitory to acid secretion. So most of the time, the agonists are working in tandem to go in the same direction. There is one exception to that, and that refers to secretin. So I showed you this at the very beginning to illustrate how autonomic nervous, the nervous system, the smooth muscle function, and the hormones work together cooperatively and integrate responses. So one example is actually the production of acid by the parietal cells. So gastrin is a direct secretagogue on the parietal cells, but it's not the most efficient one. The more efficient way gastrin works is actually stimulating these enterochromaffin-like cells, and those release the histamine, and that is now a very strong secretagogue for parietal cells. So gastrin goes directly, but also indirectly, through these cells and stimulating histamine. Then we have the parasympathetic innervation with the acetylcholine, and together with the stomach motility that mixes these secretions now with the acid and the enzymes, you're optimizing the overall digestion process. Now, again, I show you this schematic because this is again to illustrate that there is it has specific concrete ramification for treatment of excessive acid production. This is the target site for another very common group of drugs that are used against excessive acid production. These are these H2 or histamine receptor type 2 inhibitors, such as cimetidine or ranitidine, or better known probably under the names of Tagamet and Zantac and so forth. There's a whole bunch of those. Those drugs, they bind to these H2 receptors, but again, that is different from what we just learned about the proton pump inhibitors. These bindings are not irreversible. They are at the equilibrium, and when the drug levels drop, then you also have a unbinding of these compounds from the receptor, and therefore the effect becomes less and less. So those drugs have to be given more frequently compared to the proton pump inhibitors. But we're going to talk about this a little bit more details in the acidity workshop we have at the end of this week and beginning of next week. One last thing I want to show before we stop for today. the stimulation of acid secretion in response to food. And that here we can distinguish between three phases. The gastric and the intestinal phase stimulate acid production just by the fact that food arrives in the stomach, and later on, at least at the beginning, in the small intestine. This overall is a stimulus for acid secretion. But what is interesting is this first phase, the cephalic phase, that actually chewing, but before swallowing or swallowing itself, the taste, the smell of food, or even the thought of food can stimulate the acid secretion in the stomach. And that's kind of interesting because it's one of these examples that clearly are some kind of a long-range effect. Because if you look at food, you think about food, and your stomach starts to produce acid, that's certainly not something that is organized locally within the GI tract. And the knowledge of this goes actually back to quite some time. This was an experiment that was done at the end of the 19th century by a scientist of the name of Pavlov. What he did is, and I'm not quite sure if these kind of experiments would have an easy way to get permission to do those things these days, but at that time probably was easier. He operated on a dog by producing a fistula that allowed the dog to eat, but the food will not get into the stomach because it would drop out again through that fistula. At the same time, he produced access to the stomach in order to collect secretions produced by the stomach. And what he observed is that when the dog would eat, but nothing would get into the stomach, he still could collect increased volumes of gastric secretions that were high in acid with very low pH. So that got some insight into the regulation of intestinal or gastric secretion specifically. But Pavlov was also interested in to understand how learning works and how animals or humans could be conditioned to do certain things. So what he also did by repeating this experiment is all the time when the dog got some food, he would ring a bell and repeated this experiment over days and weeks. And eventually the situation was tested that just ringing the bell without providing any or presenting any food would also lead to release of gastric secretions. So in other words, this repeating pairing of giving food and ringing the bell created a conditioned reflex, a learned reflex, through which now another stimulus, which has nothing to do with the ingestion of meal, would cause the same effect, meaning in this case the secretion of gastric juices. So this was actually one of the very important early findings that then lead to the development of different types of learning and conditioning that developed over periods of time after that. So these are some original drawings of that fistula operated, and this is an original drawing from that work showing the way the experiment was set up. This was actually one of the very earliest Nobel Prizes in medicine that was given for two reasons. One, the insight into the understanding of gastric secretion regulation, and the other one, the new insight into pathways or mechanisms of learning with these unconditioned and conditioned reflexes.